Welcome, everyone, to the kind of Statistical Genetics BroadE Workshop. There were, I think, something like 251 attempted students, so consider yourself amongst the lucky few in the scheme of things. And really what we're going to try and do today is establish some of the basic principles associated with kind of basic statistical genetic analysis, and really try and understand how we think about more complex traits. I think when you get taught genetics in school and you go over all of these genetic principles, you're mainly taught what I would consider a more Mendelian style analysis rather than what we would think of as more quantitative measures and metrics and things like height and schizophrenia and diabetes. Those kinds of traits have a different set of considerations. So make sure that everyone, finds a seat. There's a bunch of seats in the front, which is typical, I think, for this sort of course. And there's a laptop on each table that's been preloaded with the data set that we're going to be talking through, and also configured so that the practical will run a little more smoothly. So I would encourage you to work with your partner through the course of the day, and we really will do some hands-on kind of Plink-centered activities to understand the basics of GWAS analysis, kind of QC, some of these, and basic association principles. But to start, we're going to ground ourselves in theory. And this theory is really a century old. So what we're doing right now in complex trait genetics is really the culmination of kind of about 100 years of theoretical work, 100 plus years of theoretical work about how we get normal variability in the population and how that is derived from genetic factors, at least at some level. So I'm going to provide this kind of introduction to the biometrical model. There will be a little bit of math, but most of it will be conceptual, so hopefully it's not too scary and horrible. But the math is important as well because it really has strong implications for the kinds of ways that we think about analyzing the data and so on and so forth. So let's just start with our good old Mendelian genetics. So this is the genetics that you all learned in school, and this is the genetics that everyone thinks about when they think about kind of trait genetics. And we have our our kind of yellow pea that's got a smooth skin, and then our wrinkly green pea, and the yellow and smooth are the dominant traits, and the wrinkly ones are the recessive traits. And we make this first generation, and it looks all yellow smooth with crossing these two kinds of peas. And then we get into the breakdown of the cross within the self-pollinated cross in that F1 generation. Now, the favorite fact of mine about Mendel is that because he was doing this work in the kind of mid to late 19th century, the concept of error and error variance and kind of estimation was absent, and so all of his experiments came out perfectly with respect to what he expected the genetic laws to be. So there were no mistakes, there was no error in his data set whatsoever. And it's admittedly unclear whether it was Mendel or whether it was some of the other members of the monastery that were kind of fixing the numbers to get precisely where the expectation was. But the laws, still hold, and we still have that big girding of Mendelian genetics that kind of really defines a lot of the basis of our theory for thinking about genetic transmission within families and those sorts of things. However, we also have codominance. So if you go back and look at some of the other traits that we have, you have these white flowers and these red flowers, and now we have washed out flowers. That's okay. Is it too dark? A little lower, a little brighter, something like that. We're recording, so please bear with us while we're experiencing technical difficulties. I won't make the technical difficulties sound for you though, because I find that noise annoying. All right, so we have this codominance of white flowers and red flowers, and we cross them together, and we get a bunch of pink flowers. And if we cross the pink flowers together within themselves, we get this white flowers a quarter of the time, pink flowers half the time, and red flowers a quarter of the time, recapitulating what we have. And then similarly, if we cross white with pink, we get half pink and half white. So this is basic, discrete trait genetic theory that you should have all learned when you were in school. And that was more or less like all you got. You got a little bit of dominance, you got a little bit of recessive, you got a little bit of codominance, and then that's kind of where we left off. But that being said, there was another series of observations that were occurring at around the same sort of time, maybe a little bit later than Mendel. And Francis Galton is kind of one of the most exquisite examples of this, where he was looking at the kind of deviates of the children are to those of their mid-parents as 2 to 3. So what that really means is that you have mid-parental height here, okay, so this is that line, and then you have what the children look like based on the mid-parental height with this line. So this is where we get the term regression. This was the beginning of the concept of regression, and the idea was that if you have mid-parent heights that are very, very tall or very, very short, then the children tend to be shorter and taller than the rest of the population on average, but they also drift towards the mean a little bit. And so this drifting towards the mean is what we refer to as regression towards the mean. And it's a really clear articulation of this idea that these more quantitative phenotypes that are continuous in the population, things like height, are actually also being influenced by genetic factors, and the extent to which they are being influenced by genetic factors can be estimated by looking at what the parental values are and then what the offspring value is. And then similarly, that got extended through to the twin method as well that Galton almost got to and recognized that there was information in twins about these sorts of things as well. And that's where our kind of modern heritability studies come from and all of those concepts. But we still have this problem that we have on one hand this discrete characteristic Mendelian flavor of genetics. Okay, so we've got like white flowers and pink flowers and so on and so forth, or wrinkly peas and smooth peas and all that kind of stuff. But we also, on the other hand, have this continuous variability that seems to be more similar in families. Tall families tend to have tall kids and short families have short kids and so on and so forth. And then there was a little bit further work on this kind of observation. So this is a paper from 1915 from East that looked at the corolla length of the tobacco plant, okay? And this is another of this kind of long series of breeding experiments done in plants and animals to really understand the nature of genetics in a much more controlled and experimental fashion. And what they did was what East did was he took plants at the top end of the length of the corolla and at the bottom end of the length. And so he kind of isolated these two extremes, and then from those isolated extremes, crossed them and got an F1 generation. So in that first generation, what we see is that the kind of variability is about the same as we see in the extremes, and that they're right in between the extremes. Okay, so we got the two poles here of very short and very long corolla length, and then we get F1 in the middle, and then it crossed within F1, so this is self-pollination within F1 to get to F2, and you see a much broader distribution of variability. Okay, so here we have approximately the same mean between F1 and F2, but we have very different variability. Okay, so there's a lot more variance going on here in F2 than there is in F1. And this is consistent with this concept of polygenic inheritance. Okay, so we've got lots of genetic factors that increase length, lots of genetic factors that decrease length. We basically heterozygose all of those genetic factors in this F1 generation. And then in the F2 generation, what we see is much more variability. And then we can selectively breed across different points within this F2 generation, within itself, and reestablish new means for the distribution of corolla length. And so that is consistent with this idea that there are a great many genetic factors that are conspiring together to give rise to this overall distribution of corolla length in the tobacco plant that we see experimentally. So in 1918, Ronald Fisher wrote, I think, what is probably the single most important paper with respect to this and many other applications. So this was a letter to the Royal Society that is titled The Correlation Between Relatives on the Supposition of Mendelian Inheritance. Okay? So here we have these two observations, this Mendelian inheritance on one side, and then this continuous, correlated family members on the other side, and how do we reconcile those? And that's exactly what Fisher did, is he said, well, in the event that there is a lot of little genetic factors, we would get an emergent normal distribution, and from that emergent normal distribution, we can then see this correlation in families. So he defined and kind of explained this pattern that we were seeing here. He provided the theoretical basis for this pattern, and then also introduced the concept of variance. So really, in terms of a paper, that's pretty good. So it's kind of groundbreaking work in genetics to reconcile the two major camps, because you have the Darwinists who are like continuous traits, evolution, and all that kind of stuff. And then you have the Mendelians who are like discrete traits, everything's inherited as a yes or no, so on and so forth. And the vestiges of those two kind of communities still exist in the genetics community today. You can see clear tradition lines of people that have a much more Mendelian view of the world and people that have a much more quantitative genetics view of the world. Well, Fisher tried to set about reconciling these two camps, and I think has brought a lot of these people together as a consequence, but there's still vestiges and holdouts and so on and so forth. And so he invoked something called the central limit theorem, and I'm not going to kind of define it. I'm just going to show it to you in a series of dummy pictures thinking about tossing coins. So whenever you have a statistician before you and he's going to start talking about methods, coin tossing is very high on the likelihood of things that you're going to hear about. The rolling dice is also popular, but coin tossing is nice because it's binomial. And so if we just have one coin and we do a single coin toss, we get half the time heads and half the time tails. But if we start to do a few more coins, so if we do 2 coins instead of 1, we'll get a quarter heads, half heads tails or tails heads, and then a quarter tails tails. And then we can continue on and do more coins and so on and so forth. So by the time we get to 11 coins, we start to see this normal distribution beginning to poke out. And if we do an infinite number of coins, we will get this normal distribution. And so that's the kind of graphical intuition behind the central limit theorem. The idea that when many small things come together and you're summing up many, many, many small effects, your emergent distribution is the normal distribution. And so that's why we talk about the normal distribution a lot in the context of statistical analysis and thinking about traits that are influenced by a wide array and wide diversity of different effects. And so here would be our kind of plot of something like heads minus tails overall for a great many outcomes and so on and so forth. So that's how we get this concept of a normal distribution from many genetic factors conspiring together. So they really all sum together. They're all, itty-bitty effects, and then we get this nice normal distribution. You may ask yourself, well, what about disease traits? We have, diabetes. You have diabetes or you don't have diabetes, or you have schizophrenia or you don't have schizophrenia. How do we think about those kinds of traits as well? Well, before the, all of the kind of neo-Darwinist reconciliation and so on and so forth by Fisher, Pearson and Lee in 1901 made an observation about something called the liability threshold model, okay? And this model is basically one whereby we have a discrete trait in the population, like schizophrenia or diabetes, or in the instance, the example that they used was horse coat color, okay? So you think of horse coat color as a discrete trait. There are gray horses and brown horses and black horses and so on and so forth. But they said, really, it's not like there's a magical discrete gray and a magical discrete brown and so on and so forth, but what you have is a continuum of all the way from light coat color to dark coat color in horse color. And so we really have this underlying liability of coat color that exists, or this underlying liability of schizophrenia or diabetes or what have you. And then above some threshold, or between two thresholds, you're going to present with a particular phenotype. And this works very well, especially when the central limit conditions are met. So when there are a great many small genetic factors running around the population, which I think we now know unequivocally to be the case for the vast majority of complex traits that we look at, then this works. This normal distribution is a pretty good model. All models are wrong, but some are useful. This is pretty far out into the tail of useful model. It's a very, very good guide. It's not ironclad, but it works extremely well in a great many different circumstances. So let's talk about now moving from this composite idea into an individual genotype space. Okay, so we have a genotype that influences a trait in the population. So here we have a SNP, just going to be a biallelic marker, that has some effect. It increases the mean in some individuals, their kind of mean expected value. It decreases them in other instances. And so what Fisher kind of put forth in this initial paper is the idea that we can have our 3 genotype classes, because we're diploid, and we kind of figured out that we were diploid quite not, before this happened. And so we have our 3 genotype classes of little a, big A, and then big A. And then we can have mean values of these things, okay? So this is under a scenario where there's no dominance, that the individual variant itself is acting in a purely additive way, okay? So that is to say, we have a mean of minus A here, a midpoint of 0 for the other class, and then plus A for the big A, okay? So that's the effect that having the 3 different genotypes will have on your trait value. So if you have little a, if you take all of the individuals with little a, they, on average, their mean will be at minus A. If you take big A, their mean will be at basically 0, though there's a little correction for the overall grand mean, and we'll get to that in a moment. And then big A will have a plus A of the effect. This is basically thinking about the beta on a regression for that individual SNP. Same sort of concept, and that's exactly the things that we're going to do with respect to the practical a little bit later on today. Now, we can also have dominance. So some things may not be acting purely additively, like, wrinkly pea skin or whatever. Take your pick. Lots of examples of things like that. So when we think about the genotypes in the mean space, so here we would have, this would be the distribution of little a, big A, and little a over here. And the different means that exist are the genotypic mean values for those. The grand mean will be the mean across all 3 of these considered together. That would be the whole total population. And so when we introduce dominance, what we're doing is we're moving this heterozygote class towards one or the other. Now, you can have overdominance. It's not super common, but it does, happen. There are experimental conditions whereby you see a higher trait value mean for an individual that is heterozygous than either homozygote. But by and large, most of the things that we seem to be able to detect at this point in time act additively and almost completely additively, although those are the things that we identify most rapidly. And so we have this minus A, and rather than having this heterozygote class be smack dab in the middle of little a, and big A, instead it moves around. And that dominance deviation also adds to variability in the population under some circumstances. So where these genotype classes end up contributes some of the overall variance that we look at when we see this population. I know it's all theory and math, and it's kind of dull and boring, but it's going to get worse, so, there's that. In reality, what we're trying to talk about here is the mean of the population and the means of these genotype classes. And so you remember your mean, sum up all your observations and divide by n, and we get this nice distribution. And then we can also do the same thing for variances, where we're looking at the deviation from the mean for each individual sample and looked at that squared deviation overall. And that gives us the idea of how much stretch there is in the population, how much variability there is. And then there's this concept of covariance, where you have the relationship between 2 phenotypes. So you have this, kind of x equals y, some sort of strength of relationship between 2 variables. So those means, variances, and covariances are what we use to try and understand what the effect of genetics is in the population. And so if you go and read the twin literature, the family and adoption study literature, almost all of that is concerning itself with observing what the means, the variances, and in particular, the covariances between relatives looks like. Because as we are more similar genetically, so I have a sister, I share on average about half of my genome with my sister, that makes me a hell of a lot more similar to her than almost anybody else in the population, except for my parents and my other siblings, is in reality. And so we should be a lot more phenotypically similar for all of those traits that are controlled by genetic effects. And so when we talk about heritability, that is what we are trying to measure, is how much more phenotypically similar are you going to be conditional on being more genetically similar. So now let us work through, and this is the really ugly math, and I'm sorry about that. So now let us think about the contribution of the particular quantitative trait locus, that QTL, that SNP that has an effect in the population on the mean. Okay, so that's what we're going to try and understand here. And so we can think about this as cholesterol level in the population. You can think about it as height. You can think about it as any sort of trait. And if we want to think about this in the context of schizophrenia, we can think about it as a contribution to liability. So there's a liability component here that we can think about as well. And so we have these genotype classes, this big A, little a, and little a. Under Hardy-Weinberg, our frequencies are p squared, 2pq, and q squared, where p is the frequency of the big A allele, and q is the frequency of the little a allele, which is equal to 1 minus p. And we all know our Hardy-Weinberg of, kind of the probability of the genotypes under a whole bunch of assumptions, most of which are mostly met if you look at a homogeneous population. And so our mean, our grand mean of the overall population is this genotype mean times the frequency of the genotype, plus this genotype mean times the frequency of this genotype, plus this genotype mean times the frequency of that genotype. And that gives us this kind of multicolored equation down there. And that ends up as being basically a times p minus q plus 2pqd. These aren't essential. If you're trying to do statistical genetic modeling and those sorts of things, these become more useful. If you open up most statistical genetics textbooks of some description or other, they will have a description of the biometrical model, because this is what is explaining variability in the population. And that's what we are trying to capture when we are doing these statistical genetics association kinds of analyses. We're trying to explain the overall variability that we see in the population. So here's our grand mean, and we need to define our grand mean so that we can understand what our variance is. Okay, so we're now going to talk about the variance, and this is where the action is. This is where, kind of statisticians get very excited because they're trying to explain variability. If there's no variability, then there is no ability to test. Okay, so variance equals something to explain, something to describe. We have error variance, where you make measurement errors or, whatever. You can think about people, measuring people and having a slightly wrong yardstick or something like that. But we also want to explain genetic variance. So what are the genetic differences in the population that yield phenotypic differences in the population? And so when we talk about explaining variance, it's that kind of variance that we're interested in. And so here, once again, we have our genotype classes of big A, little a, and little a. We have the various effects. Our average, our mean for the population of individuals with this big A is this. Our mean is this for our big A, little a. That's that dominance deviation thing. And then for little a, it's minus a. And then again, we have these frequencies. And so what we do is, to calculate the variance, we take this genotype mean and we subtract off the grand mean. So if you remember your variance formula, it's, each individual observation minus the mean squared divided by the total number of observations. Well, here we're dealing with the population, so it's simply this genotype class's mean minus the grand mean m, quantity squared, times the frequency of these observations, which is calculated based on the allele frequency at this particular genotype class. So we have a minus n squared times p squared plus d minus m squared times 2pq plus minus a minus m quantity squared times q squared. And that now is a closed form solution for the amount of variability in the population that is due to this QTL and this QTL alone. So when people talk about how much variance is explained by this locus, this is the kind of equation that they are invoking. This is the way that they are thinking about that from a purely theoretical point of view. There are experimental empirical methods, but for all intents and purposes, they generally agree with the theoretical model more generally. So this is our variance QTL. And so it represents the heritability of x at this locus is the variance of the QTL divided by the total variance. Okay, so if we have something that's got a mean of 0 and a standard deviation of 1, and so the variance is 1, and I have a QTL that explains 0.01 variance units, 0.01, I plug in all my equations, I get 0.01. I've now explained 1% of the variance of the trait, and that represents a 1% heritability contribution. When we talk about percent of heritability, rather than dividing by the variance of the total trait, we divide by the estimated variance due to genetic factors. So we use our heritability estimate there for the variance rather than the overall variance in general. So here's our variance of x. We can break this down, with our M and go through all of these ugly equations that aren't that exciting, but I want to draw the kind of distinction about additive genetic variance and then dominant genetic variance. And the reason that this is important is a lot of the conversations around this model and many others center on this question of, is it additive, is it dominant, how nonlinear is it, so on and so forth. And most of that conversation resolves to the fact that for all intents and purposes, additive effects represent the main effects of those individual genotype classes, and then dominants are the kind of deviation from that additivity. Under most scenarios of dominance, additivity captures most of the variance. Okay, so while it may actually be acting in a dominant fashion, additivity does a pretty good job of capturing a lot of that variability. So while in truth it may be dominance, in statistical space, most of it looks like it's additive. Kind of somewhat inside baseball observation. So that's kind of like the end of the math bit. Does that make sense? Does anyone have any questions? This is our biometrical model. This is the kind of basic principle that organizes the vast majority of the kind of current statistical quantitative genetics literature. So we've got a century of this, and all of the things from the kind of Peter Vischer GCTA analysis straight the way through much of the association testing relies on this conceptual theoretical framework. It's a really integral component to what we see more generally. So what about the genome? Just a few words on the genome. It's kind of, most of you are just as familiar, if not more familiar with this than I am. So we have our Watson and Crick, 1953, our various bases, A, C, G, and T, yada, 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 just a kind of quick review. We have sequencing. A little bit of it gets generated here. There may be some people here that were somewhat integral to these projects, just at a guess. $3 billion and, 3 billion and change bases, more or less. And then we get into other concepts of, we have our chromosomes and all of these sorts of things. This is just to refresh your memory and be thinking about these are the sorts of things that we're going to be looking at. In particular, the single nucleotide polymorphisms. And it is really this catalog of variation from the 1,000 Genomes Project, but also from the HapMap projects and the SNP consortium and all of the forerunners to these activities about the 30-odd million SNPs that we look at that we see as variable in the population and are reasonably common enough to observe from sequencing. It is expected that the vast majority of the genetic differences that we observe can be captured by the SNP variation. Not all, copy number variation, inversions, all those sorts of things will contribute to variance in the population. But SNPs is where a lot of the action is. And the reason it's where a lot of the action is because it is easily the most common form of genetic variation that we see in the population. And it certainly is explaining a reasonable chunk of what we're observing. And they really do contribute to a whole variety of things, from height and weight to how we behave, whether we're extroverted or introverted, whether we're kind of excited to go out at night or want to stay in. Those sorts of things have, at least at some level, some genetic influences on them more generally. So we've got our SNPs, and this is just the idea of the SNP here is that we have 3 individuals in the population. We have this AA at this position, this AC, and the CC at these positions. These are the single nucleotide polymorphisms. They should all just be nice, easy refresher. You went through the math, and now it's like, genetics, this is easy, hooray, we know all this is great. But it's just good to make sure that we're all on the same page. And really, this mutation arose at some point. If you want to talk about mutational differences, talk to Shop. That might be a good idea. He might have some ideas about conservation across different populations. I always get uncomfortable talking about some of these concepts in these sorts of environments where there are people that study just this part of the whole landscape when I have to do it in a dilettante sort of fashion. And then just a few definitions, because really when we're gonna use this terminology just to make sure, I know it's tedious and boring. So we have our haplotypes, so this represents an individual. Our haplotypes is the series of bases, one basically stretch inherited from your mom, one stretch inherited from your dad. And then we have genotypes, which are the 2 variants you have at a single base pair position. So our SNP is those 2 genotypes, that genotype concept. And then we have individual alleles here. So we have kind of allele 1 and allele 2. And then we might have some phenotypes, some affection status, some quantitative trait. And we're really going to try and link genotype to phenotype. That's going to be the name of the game. And a lot of the way that we're going to do this is just mean differences or linear regression. We can do it in chi-square. There's a lot of diverse statistical methods, but the association practical is really going to show us how exactly we go about doing this in a very hands-on fashion. So our nice little simple regression model. Do you have a copy of the big A allele, yes or no? If yes, do you have a higher mean than if you don't? On average within that population of carriers, and so on and so forth. Just a few slides on technology. Where does this data come from? Most of the data that we're going to be talking about here is the genome-wide association data. Although if you went to MPG yesterday, we need to call this GWAS now. So really the common variance is what we're trying to look at. And the basic technology is that there's kind of basically a primary sequence that matches a known variable site in the population slapped on a slide that'll fluoresce depending on what gets annealed to it. That fluorescence then gets turned into a kind of intensity 2D plot. I'll show you a picture of that. So here's the AFI chip. We've got our restriction enzyme digestion. We're going to kind of ligate some adapters, and then we're going to reduce the complexity, wash it over the chip, and then we're going to get these different intensities for the different genotype classes that we see more generally. I think George Grant is in the audience, and he can also tell you a lot more about this kind of process than I can, among others. And this is, what one version of a set of Illumina chips look like. There are a bunch of these different genotyping platforms that exist across the community, and we're kind of constantly redesigning a lot of them to make them kind of new and improved and so on and so forth, and capture more genetic variation or lowering the cost. But this is the kind of 2D intensity that I was talking about. So you'll get like, a set of intensities for one cluster here, and one cluster here. And the idea is you meant to draw circles around them, okay? So we do this in an algorithmic manner. And those algorithms have evolved over time. There are a whole diversity of them. You could spend an entire lecture on genotype calling. I'm not sure that would be of the broadest appeal, but it is very important. And it doesn't always work. So here's a slide that Jeff Barrett made from the Wellcome Trust Case Control 2007 paper, whereby you can see well-behaved clusters. So here we have 3 nice distinct clusters. We're all very happy. And then sometimes you get like muck here, and then you get maybe like 6 clusters, and maybe this site is kind of variable in a copy number way in the population, in addition to having a SNP genotype in general. So, these things are legitimate concerns and will cause some amount of genotyping error. And we can capture this genotyping error by QCing our data, and that's really what we're going to turn to now. So that's like the biometrical model, just a little introduction to refresh your memory on all of the various terminology pieces that we're going to be using over the course of the rest of the day. We're going to start with introduction to QC and a practical component from Wernery, introducing Plink, how to use Plink, all of the various bits and pieces that come into that. Then we'll have a little break so we can stretch our legs and make sure that we're still awake. Then we'll teach the concepts of population stratification and have a practical component on that, and we'll see how that goes, and then have a little other break. And then we'll do some association testing and quantitative traits, and this is the kind of the last major scientific session. This will be hands-on. Use the computers that you found on the desk, so you should all kind of pair off, and so it should be 2 to a computer. And we're going to run through commands, we're going to look at output, we're going to plot things in R, we're going to use the command line from the terminal from the OSX. If you have any questions or you get stuck, the moment you get stuck, put up your hand. There will be people in the room who have run through the practical and can help sort out your problem and understand what's going on with respect to your data. If you have any other questions, don't be shy. This is an educational workshop, and the whole idea here is that you are engaged in the learning process, and so you can understand what's happening in particular. So that's the course for today. Then we'll finish with a little bit of theory slides. If we run long on time on the practicals, we'll kind of selectively axe one or the other of the talks as we see fit. So any questions at this point in time? You've got the biometrical model in your mind. There are lots of small genetic effects that conspire together to give us this normal distribution. And that's what we're going to try and understand. Why is there variability in the population? Where does it come from? And what can we understand about it? And with that, I will hand over to Wernery.